All right, so we're recording, and we're going to go over the digestive system, which is part of material on lecture test 4, which is going to cover the digestive system and the urinary system. So first thing would be anatomy. The anatomy of the digestive system is divided into two groups of organs. The organs of what is called the alimentary canal or gastrointestinal tract or GI tract. These are the organs that ingest the food and proceed to digest the food and then absorb it and eliminate the waste. The accessory digestive organs are the helper organs that are going to help the GI tract organs do their job. So in lecture and in lab, you should be able to identify the anatomy of the digestive system. So we start at the mouth. So this is a canal. So you should be able to trace the path of food from the mouth all the way to the stomach and intestines. So from the oral cavity, food is digested actually in the oral cavity. That's where digestion starts by the actions of enzymes in the saliva and the physical action of the teeth and saliva together crushing the food. The food is swallowed, so it's going to go into the oropharynx, from there into the laryngopharynx, where it will be diverted into the esophagus, which is the tube that is going to take the food to the stomach. The stomach continues some of the digestion of the food via acids and a little bit of digestive enzymes. That pre-digested food, which now is called the chyme, is going to be going into the small intestine, and the very first part of the small intestine, about 6 inches of it, is called the duodenum, and that is where the bulk of digestion will take place. And absorption, actually, digested food will continue on to the next portion of the small intestine called the jejunum. And again, some absorption takes place in the jejunum. And from there, it will travel to the last portion of the small intestine, which is the ileum. So the ileum brings the food, or that point, it will be the waste, into the large intestine. First part of the large intestine is the cecum, right here. And from there, the food is going to go up. Or I should say, I shouldn't call it the food anymore. The contents will go up into the ascending colon, which then is going to cut across to the other side of the abdominal cavity through the transverse column. Then it will be moved down through the descending colon, which then leads to the sigmoid colon, the rectum, and the anal canal. So that is the path of the GI tract. Accessory organs will be the liver with the gallbladder, pancreas, you can see a little bit of the pancreas right there, salivary glands, you can see a huge salivary gland here, there should be another one down here. I think that was it for the salivary glands, for the accessory organs. All right, so you should be able to identify these organs in a picture. The digestive process involves taking in the food, which is ingestion. In the oral cavity, food is masticated or chewed, and that is physical digestion of the food. There will be also some chemical digestion at that point also. The movement of the food through the GI tract involves peristalsis. The mixing of food throughout the GI tract is called segmentation. Deglutition is another word for swallowing. Digestion involves secretion of enzymes. Digestion is chemical and mechanical. So there will be absorption of the nutrients and then elimination. This PowerPoint depicts the two different types of movements that take place in the GI tract. Peristalsis is a movement that propels the contents along the tract, and you can see how the bolus of food is being moved from this position to the next to the next. Segmentation, on the other hand, is a movement that mixes the bolus of food. So you can see how these areas are being mixed. So the food is moved up and down. or the contents are moved up and down. All right, when it comes to the histology of the alimentary canal, pretty much starting at the esophagus. The histology of the esophagus, the stomach, and the intestines is very similar. You can divide the layers, the histological layers, into 4 distinct parts. The mucosa, which is the innermost layer. Below the mucosa is the submucosa, muscularis externa, and serosa. So what we have here is a cross-section of the intestines, of the small intestine. So the mucosa is going to be this entire area right here. This entire thing right there is the mucosa. This is the lumen, the interior of the GI tract. And it is showing you the components of the mucosa, which we're going to follow here in just a second. Below the mucosa is the submucosa, which is this gray area right there. Below that is the muscularis externa, which is this thick layer of the smooth muscle right there. And then the serosa or adventitious, this thin layer down out here. The mucosa itself is made of the mucosal epithelium, which is the layer of epithelial tissue that lines the GI tract, and for the most part is going to be simple columnar epithelium. The lamina propria, which is a thin layer of connective tissue, and the muscularis mucosa, which is a thin layer of smooth muscle. So again, the mucosa will be made of the simple columnar epithelium, which is this little pink line right there, the lamina propria, which is this yellow area right here, and the muscularis mucosa, which is this thin layer of muscle right there. Notice that the epithelium lining the GI tract, especially starting at the stomach, is going to make folds. It's going to fold, creating these finger-like projections right there. This is the intestine. This is going to be the villi of the intestine. But something similar to that will be seen also in the stomach. And then it also folds downward, down here. So there's downward folds, which are going to be called intestinal glands or crypts. Below the mucosa is the submucosa, which will be this layer right here. Muscularis externa, which are the layers of muscle, and then the serosa, which is a thin layer of connective tissue outside. And by the way, these are pictures from a model in the lab. This is a side view of the model. This right here is the mucosa, the epithelium of the mucosa, which is made of simple columnar epithelial cells. And this is showing you the glands going inward. And these structures have very different types of cells right there. This is the thin layer of epithelial tissue right there. This is the muscularis mucosa. This entire gray area here is the submucosa, a thick layer of muscle, the muscularis externa. Now, notice these yellow lines here, which you can also see right there. Those corresponds to nerves. So there's a plexus, a network of nerves in the submucosa called the submucosal plexus. And the submucosal plexus begins in the intestines. So the esophagus and the stomach do not have the submucosal plexus. Let me show you here. Again, this yellow network of nerves is the submucosal plexus, which we begin to see in the intestines. The muscles. The muscularis externa is a thick layer of muscles. For the most part, we're going to have 2 layers of muscles going at 90-degree angles of each other. So we're going to have a circular layer and a longitudinal layer. There are exceptions. This is a smooth muscle, by the way. The stomach has 3 layers instead of 2. And in the esophagus, the first 1/3 of layers of muscle are going to be, this is wrong. It should be the upper 1/3 layer is skeletal muscle, not a smooth muscle. So that should be skeletal. I'll check to make sure PowerPoints are okay. Ours may be. These are not exactly the PowerPoints on eCampus. All right, so at first the esophagus is a skeletal muscle and then it changes to smooth muscle. Also notice, I don't know if you can see this yellow area right there that represents another plexus. And in this case, the plexus is called the Myenteric Plexus. So this right here is the plexus of nerves that controls these skeletal smooth muscles right there. And again, that will be the Myenteric Plexus. Okay, so that's showing you the Myenteric Plexus there, the Submucosal Plexus. So the Myenteric Plexus is going to control those layers of the smooth muscle. And this is going to be autonomic nervous system control. So some of those nerves are going to be sympathetic nerves, others are going to be parasympathetic nerves. The sympathetic nerves are going to inhibit the muscles. The parasympathetic are going to stimulate the muscles. So in general, sympathetic stimulation of the GI tract inhibits the secretions and the movement of the muscles of the GI tract. Parasympathetic stimulation of the GI tract stimulates secretion and movement of the GI tract. And then the serosa, which is essentially the visceral peritoneum. All right, so when it comes to regulation of the GI tract really has two responses to stimulation. One response is secretions. Secretions of what depends on the organ, but it could be secretions of mucus, for example, in the esophagus. It can be and even the intestines. It can be secretion of enzymes in the intestines. It can be secretion of acids in the stomach. So this is one of the responses, is secretion. Or hormones, that will be something else, hormones. The other one is movement. Peristalsis and segmentation. All right, sorry about that. Okay, so those are the two responses to nervous stimulation of the to stimulation of the GI tract. That response can be due to what we call lung reflexes, meaning the central nervous system, via sympathetic and parasympathetic stimulation, innervating these organs, glands, and muscles, and make them secrete or move. And that would be, parasympathetic is going to stimulate, sympathetic is going to stop. Again, parasympathetic stimulates, sympathetic stops. And that's what we call the Cns Lung Reflexes, because it's coming from the brain. Local control, however, are going to be reflexes. That's going to be localized stimulation due to localized arrival of food and other stimuli that appears locally. So there are two responses. One is local. The other one is coming from the central nervous system, which we call the lung reflexes, is CNS control. So oftentimes, and more, you may hear about the gut brain. The gut brain refers to this local control right here, is this ability of the GI tract to have its own little network of nerves that can be stimulated locally without the help of the brain. All right. So there are three types of neurons that reach the GI tract. We're going to have the sensory neurons, which are going to sense changes in the GI tract. These are going to be receptors, chemoreceptors, and stretch receptors. Then we're going to have the middlemen that connect the sensory neuron to the motor neuron, and the motor neuron is the one that stimulates secretion and contraction. It is the motor neuron that can be coming from the CNS, or it can be local. Okay. All right, so the sensory neurons are going to be interoreceptors are essentially two types of receptors, chemical receptors and mechanical receptors. So the chemical receptors are going to sense two things. They're going to sense pH. They're also going to sense undigested food. Whereas the mechanical receptors are going to sense a stretch of the tract upon the arrival of food. The motor neurons are going to activate the muscles of the GI tract, muscularis externa, muscularis mucosa, and the glands of the GI tract. Okay. The ENS refers to the local control. The local control that is also going to control the GI tract via movement. And actually, it's not going to be talked about much at all during the PowerPoints, but there's also control of blood flow in the area, local control, so that when we're ready to absorb the food, blood vessels in the area will begin to dilate to take in more the nutrients. Okay, yeah, we talked about interoreceptors, which sense the changes inside the GI tract, chemical changes and mechanical changes. There are also exteroreceptors that are going to stimulate the GI tract via the central nervous system. And that refers to things like the sight of food, the taste of food, the smell of food, are going to, via the central nervous system, stimulate the GI tract. And the central nervous system is going to act on the GI tract via the parasympathetic nervous system, which uses the vagus nerve, which is cranial nerve number 10, to stimulate contractions and secretions in the GI tract. On the other hand, the sympathetic nervous system is going to inhibit contraction and secretion, and it's going to constrict blood vessels also. Remember that from previous chapters. The GI tract is one of the peripheral organs that are whose blood vessels are going to be constricted with sympathetic stimulation. Okay, so this picture right here is a good summary of control of the GI tract. Give me a second. All right, so let's see. So we start with the stimulus that is going to turn on the receptors. And notice that here, the stimulus are going to be external stimulus, sight, smell, taste, even thought of food for that matter. That is going to work through the central nervous system. And through the central nervous system is going to be lung reflexes. Now we're going to number 2. Is going to act on the nerve plexuses. That would be your myenteric and submucosal plexus. The effector organs are going to be smooth muscle and glands. And now the response is going to be contraction and secretion. So peristalsis, segmentation, and secretion of digestive enzymes, mucus, pH, acids, etc. So this will be the lung reflex. The other stimulus will be an internal stimulus, interoreceptors, via chemoreceptors, osmoreceptors, mechanoreceptors. Chemoreceptors and osmoreceptors are going to detect the arrival of food, change in pH in the food, and the mechanoreceptors will change in pH in the GI tract, in the stomach, for example. The arrival of food is going to buffer the acidity of the stomach. The pH is going to go up. Once food arrives, the organ stretches, and that's going to be detected by mechanoreceptors. So these local interoreceptors are going to stimulate, again, the submucosal plexus and the myenteric plexus, which in turn are going to stimulate the effectors, smooth muscles and glands, and that's the response is going to be a contraction and secretions. So this is what we call the short reflexes versus the long reflexes. Now, notice something though. These interoreceptors are also going to communicate to the central nervous system and increase further long reflex stimulation. So the nervous system, the GI tract is getting or receiving stimulations from the CNS and locally. So this basically summarizes how all this works. So which plexus is responsible for smooth muscle contraction? Which plexus is involved in glandular secretions? Well, the answers will be, we have two plexus, myenteric and submucosal. So myenteric is the one in the muscularis externa. Submucosa is the one right below the mucosa, so mucosal plexus. And so that's the one that's going to stimulate glands. All right, so other regulation. Okay, so we're talking about regulation via the nervous system. So the nervous system is going to use neurotransmitters. The parasympathetic nervous system uses acetylcholine, and this you should know from last semester. So the neurotransmitter acetylcholine is going to stimulate the GI tract. On the other hand, the sympathetic neurotransmitter is norepinephrine. So norepinephrine is a neurotransmitter that is going to inhibit the GI tract. These, however, are CNS neurotransmitters. The neurotransmitter that is used by the nervous system of the GI tract, which is called the enteric nervous system, or ENS, is serotonin. So this is the neurotransmitter of the ENS, and it's going to be a stimulatory neurotransmitter. Serotonin is also a neurohormone, which is going to be secreted into the blood by enteroendocrine cells of the GI tract. So just like norepinephrine is a hormone and a neurotransmitter, so will serotonin be a neurotransmitter or a hormone if it is secreted into the blood. The effect will be the same, stimulation. So we talked about serotonin as a hormone, in this case neurohormones, and it will be a stimulatory, whereas norepinephrine is going to be inhibitory. And it can also be a neurohormone when it's secreted by the adrenal medulla. Okay, other hormones secreted by the hormone-secreting glands of the GI tract, which are called enteroendocrine cells, are going to be gastrin, which is an stimulatory, secretin, which I'm not going to call it a stimulatory and inhibitory because it has kind of both. It stimulates parts of the GI tract, inhibits other parts of the GI tract. So these are hormones. Now, there are other chemicals that act on the GI tract. Paracrine chemicals are those that are secreted locally and act locally. Remember, hormones travel through the blood and act on organs or tissues that are far away from the place where they were secreted. These paracrine secretions are local, and the main one is histamine, which is going to have a stimulatory effect. And we'll go more into exactly what these chemicals do. Okay, so the next PowerPoints are going to look at mesenteries, and a lot of this is lab also. So I am tempted to just skip all these, and you're going to get this information from lab. Make sure you understand that you also need to know it for lecture. Do know the retroperitoneal organs, like the duodenum, pancreas, rectum. Do you know the mesenteries? Okay, just thought I had a better view of mesenteries. I guess this is going to be it. Okay, so the mesenteries you need to know will be greater omentum, which are going to deal with the large intestine, the mesentery proper, which puts the small intestines in place, lesser omentum between stomach and liver. Okay. All right. Okay, so there's the lesser omentum between the stomach and liver. The greater omentum has been cut right there. Falciform ligament also. I knew there was one more. I can see it here. So the falciform ligament will be another one that you will need to know. And these are the same that you need to know for lab anyway. Okay. Again, I'm not going to spend too much time on this. Do know the dental formula. And you need to know that for lecture and lab. Different types of teeth and what they do, how many of them there are, the parts of the tooth. Okay, again, this is part of lab also. Salivary glands, know their location. So the parietal gland is the largest. There's two of them. Then we have a submandibular gland and a sublingual gland. Functions of the salivary glands is to secrete saliva, which contains enzymes that are going to start the breakdown of the starch. That is important. Definitely know that. The digestion of carbohydrates begin at the mouth due to the actions of salivary amylases, right there. Okay. Know the composition of saliva, and obviously know salivary amylase. All right, the process of swallowing or deglutition has several phases. The first phase is the voluntary phase, which is a swallow reflex, which is carried out by cranial nerve number 10, in which the bolus of food is going to be pushed against the palate and then back to the soft palate towards the oropharynx. That requires the action of the tongue and the elevation of the soft palate. And then the food is pushed towards the oropharynx. So this is the pharyngeal phase. The uvula closes so as to prevent the food from moving into the nasopharynx. So this is the pharyngeal phase. Pharyngeal constrictor muscles, which are going to be voluntary muscles, are going to move the food past the pharynx towards the esophagus. Now, the glottis is going to close to prevent food from going into the air pipe. And then the end of the pharyngeal phase is when the food is moved to the first part of the esophagus. So the very first sphincter in the esophagus is the esophageal sphincter, which is going to relax and allow for the food to go into the esophagus. And so we entered the esophageal phase. So we went from voluntary phase to pharyngeal phase to esophageal phase. The esophageal phase is largely involuntary. The 2/3 of the muscles of the esophagus are smooth muscles, involuntary muscles. So the bolus of food is going to move towards, through peristalsis, towards the stomach. The lower part of the esophagus has another sphincter called the gastroesophageal sphincter, which opens to allow for the passage of food. That's the gastroesophageal sphincter right here, actually, for the passage of food into the stomach. Now, both the gastroesophageal sphincter at the distal end of the esophagus and the upper esophageal sphincter at the proximal end of the esophagus are both incomplete sphincters in that they would allow for the bolus of food to come back where it came from. And the food can be regurgitated or throw up, which a true sphincter or complete sphincter is not going to allow for that. And we'll see a complete sphincter here in just a second. All right, so you should know the two sphincters. The esophagus is lined with a stratified squamous epithelial tissue because it's an area that is subject to mechanical abrasion. So I like this picture. It shows you a very dramatic view of the interface between esophagus and stomach and how the change of tissue takes place. So this is the stomach. So the esophagus is lined with stratified squamous epithelial tissue. You can see here non-keratinized stratified squamous epithelial tissue. And notice the dramatic change from that to simple columnar epithelial tissue that will be found from that point on stomach and intestines. And you can also see here Peyer's patches, which is a conglomeration of lymphatic immune system cells that will be found throughout the GI tract. So we get to the stomach, and it is an enlarged portion of the alimentary canal. The gastroesophageal opening or cardia is the part of the where the esophagus joins with the stomach, and the bolus of food is moved into the stomach. So the part of the stomach adjacent to the esophagus is called the cardia. Then we have this dome here at kind of the same level of the esophagus, which is called the fundus. Most of the stomach is the body, and then when the body begins to funnel, that becomes the pyloric area. Pyloric antrum will be the beginning part right there, I guess. Yeah, right there. Followed by a pyloric canal as the narrow becomes more of a canal. And then at the end is an actual sphincter, a complete sphincter called the pyloric sphincter, which is going to control the movement of the contents from the stomach into the small intestine. Now, notice that the stomach has 3 layers of muscles instead of 2. These are the layers of muscles. Notice that the epithelial lining of the stomach is folded into rugae, which are temporary folds that will be smoothed out as the stomach fills with food. The big dome here is the greater curvature, the smaller one is the lesser curvature. So you should be able to identify the parts of the stomach. All right, so this is showing you the rugae. These are non-permanent folds. And this is showing you what we call the gastric pits. Remember, we said the lining of these GI tract organs oftentimes goes inwards and create these glands or pits, which are filled with cells that are very, very specialized cells, which we're going to go here in just a second. These cells at the neck of the pit secrete mucus, which are going to protect the lining of the stomach from the acids that will be secreted by the stomach. On the other hand, at the very bottom of these pits are where we find the enteroendocrine cells close to blood vessels, because they're going to secrete hormones into blood vessels. Okay, so the histology of the stomach, the cells of the pits are going to be at the neck of the pit. We have these mucus secreting cells. Then further into this pit, we have a group of cells called parietal cells that secrete hydrochloric acid and a substance called intrinsic factor. Then we have the chief cells, which secrete an enzyme called pepsinogen. And finally, we have hormone-secreting cells called enteroendocrine cells. So these are going to be the neck cells here, secreting mucus. The parietal cells are these blue little cells here. And parietal cells secrete 2 things, hydrochloric acid and intrinsic factor. see. Okay, I just want to finish with the cells next. Chief cells are going to be these reddish cells here, and chief cells secrete pepsinogen. There we go, pepsinogen. Okay, so know the secretions of these cells. So again, parietal cells secrete hydrochloric acid and intrinsic factor. Chief cells secrete pepsinogen. Enteroendocrine cells here at the bottom secrete hormones. So let me go back and pick up on what these cells secrete and what they do. The cells here at the bottom are the enteroendocrine cells. They are going to secrete hormones. So the enteroendocrine cells secrete gastrin, which is a hormone that is going to stimulate the GI tract, the secretion and movement of the stomach. Somatostatin it's going to be a hormone that is going to inhibit gastrin and insulin secretion. Okay, so it's going to stop stomach. Histamine, also secreted by these enteroendocrine cells, is not a hormone, but is a paracrine product, which is going to stimulate parietal cells. And parietal cells were the ones that secreted hydrochloric acid. All right, so parietal cells secrete hydrochloric acid and intrinsic factor. Hydrochloric acid has many functions. It's going to kill any bacteria that is coming with the food. It is involved in the digestion of proteins. It is first going to denature the proteins. It's going to deactivate the salivary amylase that has been digesting the carbohydrates in the mouth. It's going to activate another enzyme called pepsin. We'll show how that happens here in just a second. Hydrochloric acid is made from CO2 in blood. So parietal cells have close contact with blood vessels, and the there we go, CO2 that is traveling in the plasma. Remember from last chapter, 20% of not 20%, about 7% of the CO2 was traveling as CO2. So that one will be taken up by these parietal cells, combined with water via the actions of carbonic anhydrase, and will be converted to carbonic acid. Remember, this is the same thing we saw yesterday in the lungs and how CO2 was transported. Carbonic acid dissociates into hydrogen ions, which are going to be pumped into the lumen, and the bicarbonate ions are going to be put into the blood. Now, bicarbonate ions are a buffer. So the blood, as it travels next to the parietal cells, it's going to be tremendously buffer, and the pH of that blood will increase temporarily. The hydrochloric acid is being pumped out by these proton pumps. The chlorine is being taken from the blood and pumped independently. So hydrochloric acid is made in the lumen when hydrogen ions and chlorine ions come together. There are a group of drugs, anti-acid drugs like Prilosec, which are proton pump inhibitors. And they're extremely strong drugs with lots of side effects, which are going to stop these little pumps from working. And part of the problem is that once you start using them, you cannot just quit out of the blue, because the pumps may never come back to working again. So you have to go through a process of weaning yourself from these pumps, from these medicines, and then hope that your pumps come back to work afterwards. It is given as an anti-acid, but it's a very extreme anti-acid, because rather than buffering the acids, it messes with the workings of the cell. So, the more we know about these proton pump inhibitors, the more reluctant are physicians to use them. They all those bad side effects they had were made clear a few years back. All right, so the alkaline tie is this temporary increasing age that the blood vessels traveling close to the parietal cells experience as the CO2 from the blood vessels is being removed to make HCl, and more importantly, bicarbonate ions are being secreted, which are going to buffer the blood into making it less acidic, more alkaline. All right, so that's how hydrochloric acid is made. Remember, parietal cells also secrete something called intrinsic factor. An intrinsic factor is a substance that is needed for the absorption of vitamin B12. Now, notice something. Intrinsic factor binds to vitamin B12 and then is going to travel with it bound all the way to the ilium, where B12 will finally be absorbed. So B12 is a vitamin that we get typically from proteins, from meats. So we need the acidity of the stomach to break down these proteins and release B12. Then we need intrinsic factor to bind to B12 and carry it all the way to the ilium, where intrinsic factor will be used to absorb the B12. So this is a complex process. The older we get, the less intrinsic factor we produce, and we may end up with a type of anemia called pernicious anemia, which is due to a lack of intrinsic factor. So B12 is needed to produce red blood cells, to produce all cells, but because we produce so many red blood cells every single day, the effects of lack of B12 is first detected with the red blood cells. So you should know what intrinsic factor is and what it does. You should know what pernicious anemia is. So back to these cells. The chief cells were the ones that secreted pepsinogen, and pepsinogen is the precursor to an enzyme called pepsin. Anytime you see the gen in the name of a chemical, that tells you this is a chemical that generates whatever the other word is. So this chemical generates pepsin. But pepsinogen, to be converted into pepsin, must be acted upon by hydrochloric acid. So what happens is you have these parietal cells secreting hydrochloric acid, and the cells underneath secrete pepsinogen. And as the pepsinogen travels through the hydrochloric acid begins to cut it, denature it into pepsin. Now, pepsin is a powerful enzyme that acts on other proteins. So once pepsin is being made, itself will go to other pepsinogen and begin to cut them into pepsin. So pepsin is needed for the digestion of proteins. So the digestion of carbohydrates began in the mouth by the actions of salivary amylase. And you can test that yourselves. If you put a piece of bread in your mouth and you begin to work it with saliva, that piece of bread will eventually turn into dough. So it's being digested. On the other hand, proteins are going to be, the digestion of proteins will begin in the stomach by the actions of pepsin and hydrochloric acid. Hydrochloric acid is going to denature the proteins. Pepsin will further digest the proteins. So the proteins are being broken down, and that process begins in the stomach. So the functions of the stomach can be divided into phases. And the initial phase is called the cephalic phase. And cephalic means head. And the reason why this is called the cephalic phase is because it begins in the head. It begins when we start to look at food, smell food, taste food, think about food. So this is the actions of external receptors that are going to stimulate the medulla, and via sympathetic parasympathetic stimulation through the vagus nerve, we're going to stimulate endocrine cells into secreting gastrin, parietal cells into secreting HCl, an intrinsic factor, chief cells into secreting pepsinogen, which will be cut into pepsin. Gastrin, in turn, is a hormone and is going to begin to stimulate parietal cells and chief cells in turn to increase further secretion of HCl and pepsin. Parasympathetic also is going to stimulate motility, peristalsis and segmentation. Gastrin will do the same thing. So this is the initiation of the digestion process in the stomach. This is a short phase that again starts at the stomach. Not the stomach, sorry, at the head, in the central nervous system. Purpose of the cephalic phase is to prepare the stomach for the arrival of food. So this begins to happen even before we eat, when we're looking at the food, tasting the food, smelling the food, and that initiates the secretion of hydrochloric acid and gastrin. By the time the food arrives, the stomach will be ready to begin its work. Remember that parasympathetic stimulation secretes acetylcholine, and acetylcholine is the neurotransmitter that stimulates parietal cells into secreting hydrochloric acid, chief cells into secreting pepsinogen that will be turned into pepsin, enteroendocrine cells into secreting histamines and gastrin. Gastrin, in turn, stimulates parietal cells, chief cells, histamine secreting cells. Histamine stimulates parietal cells. So if we go back here, we said parasympathetic nervous system stimulates endocrine cells, which secrete gastrin, stimulates parietal cells, which secretes hydrochloric acid, and intrinsic factor, we're ignoring that one for now, and chief cells would secrete pepsinogen, which turns into pepsin. Gastrin also, endocrine cells also secrete histamine. Okay, now, gastrin also stimulates parietal cells, chief cells, and further production of histamine. Histamine is the main stimulant to parietal cells. So this is the one that really stimulates parietal cells into secreting tons of hydrochloric acid. All right, so this is just showing you the cephalic phase. Following the cephalic phase is the gastric phase, and the gastric phase will begin with the arrival of food in the stomach. So the stimulus is the presence of food in the stomach. What the presence of food in the stomach is going to do is going to buffer the acidity of the stomach that had begun secreting hydrochloric acid. So that's going to stimulate chemoreceptors. It's going to stretch the stomach. That's going to stimulate mechanoreceptors. And now we're going to have the presence of proteins, amino acids, peptides. So that is also going to stimulate chemoreceptors. These receptors stimulate CNS and enteric nervous system. There is ACH being secreted. There is gastrin being secreted. Gastrin stimulates secretion of parietal cells and chief cells and everything. It's a whole cascade of stimulation that releases all of the secretions of the stomach. Notice that alcohol and caffeine stimulates gastric secretions. So, that may be where our costume of sometimes drinking alcohol or coffee at the end of a meal comes from. It's because somebody might have noticed that stimulated digestion in the stomach. The gastric phase is a very long phase. The pH of the stomach may get very, very low. However, if it gets less than 2, the whole process stops, and a chance is given for the pH to rise again before the whole process continues, because you don't want these acid secretions to become damaging. All right, so the gastric phase begins with distension and the arrival of undigested food. That is going to be that information will be sent to the central nervous system, which is going to respond with parasympathetic stimulation, which is going to stimulate secretions in the stomach. The distension also stimulates itself, gastric cells, to secrete stuff. These gastric cells, some of them, the intragastric cells, are going to secrete hormones that further are going to stimulate. Okay, so there's stimulation from different areas. So what is happening at this point is proteins are being digested, food is being churned and mixed. This is a crucial step, especially when it comes to digestion of proteins. And keep in mind, a lot of the nutrients that a protein is going to have is going to be bound to the protein, and you're going to need to begin digestion in an acidic environment for those nutrients to be released. So things like vitamin B12, iron, and other components, calcium, will need to be released through this acidity. That doesn't mean that digestion is completed or anything like that. This is the beginning of partial digestion, but it is very important nonetheless. All right, intestinal phase. So the next phase is called the intestinal phase, and this describes what happens in the duodenum, which is the first part of the small intestine. The duodenum is about 6 inches, very short, but that is where the bulk of digestion takes place. So it's an incredibly important area. Now, keep in mind, the stomach is much bigger than the duodenum. So the stomach has all these contents that we received. The duodenum cannot fit all that in. So what's going to happen is the contents of the stomach are going to need to be moved to the duodenum at stages. And so that's why the intestinal phase and the gastric phase continue back and forth for hours until all the contents of the stomach are digested. So this is not a sequential step, but it is a back and forth. So we're going to go into the intestinal phase about, a little aliquot, small aliquot of what's in the stomach will be moved to the intestines. And the intestinal volume, once that is digested, then it can receive the next volume. Okay, so that's why digestion takes such a long time. All right, so the intestinal phase begins with the arrival of the chyme, which is the contents of the stomach, which are acidic. They arrive at the intestine. The acidity of the chyme stimulates this next phase, and I cannot emphasize that enough, because we tend to think of the stomach's acids as being bad, and we try to control the stomach acidity, and we take antacids, and yet it is crucial for digestion to have an acidic content in the stomach, because if we don't have an acidic content, the duodenum is not going to be stimulated into beginning the actual digestion. So 2 things stimulate, actually more than 2 things. Like I should tell you the other ones. First one, acidity of the chyme, the stretch of the area, and, well, there you go, the acidity, a pH that it's 2 or below. Then we're going to have undigested or partially digested lipids are also going to stimulate the intestinal phase. So what do we need for good digestion? Lipids and acidity. That's what stimulates the duodenum into begin digestion. All right. Intestinal phase is going to the duodenum will begin to secrete hormones. The hormones have 2 purposes. One purpose is to stop gastric phase, stop the stomach. The stomach has to be stopped, because if it continues working, it will continue to send its contents into the duodenum, and the duodenum is already full. So we need to stop the gastric phase. That's number 1. Number 2, start with chemical digestion. Start to secrete the chemicals that are going to be needed for digestion. So hormones secreted: secretin and cholecystokinin. Secretin inhibits parietal cells and chief cells. Cholecystokinin further inhibits gastric secretions. So these 2 hormones are going to stop the gastric phase. we're going to see later that these two hormones also stimulate secretion of chemical digestive enzymes. The Intragastric Reflex, upon the arrival of food in the duodenum, is going to stimulate the enteric nervous system and the central nervous system into stopping the activity of the stomach. So we're going to have chemical secretions and then a reflex that is going to stop what's happening in the stomach, the gastric phase. This is the depiction of what's happening in the intestinal phase. So we have a low pH and lipids, which are going to stimulate the next phase. The signal is sent to the central nervous system, and the central nervous system via the vagus nerve is going to, the medulla is going to decrease stimulation of the vagus nerve into the stomach, because now we need to stop what's happening in the stomach. These local pH and lipids, the chemoreceptors are going to stimulate secretion of secretin and cholecystokinin hormones, which are going to travel, and they're going to inhibit gastric secretions. The intragastric reflex is further going to, as a local reflex, inhibit gastric secretions. So inhibition of gastric secretion are coming from all those places. CCK, cholecystokinin is also called, the acronym is CCK. A carbohydrate-rich meal is not going to stimulate CCK production, which is going to inhibit the gastric phase. So, which means that a carbohydrate-rich meal is going to make the stomach clear out quicker, partly because carbohydrates are not being digested in the stomach, they're being digested in the small intestine. So they're going to require quicker clearance from the stomach into the small intestine to be digested. Whereas a meal rich in fatty acids and proteins do stimulate CCK, and that is going to slow stomach clearance and is going to take longer to digest. That's why if you eat a carbohydrate-rich meal, you're hungry a few hours later because you've gone through it very quickly, whereas fatty acids and proteins make you feel full for a longer time because it takes longer to digest those contents. So I guess the best bet would be to have a meal that has a little bit of everything. Okay, so now this intestinal phase continues until that food that has arrived or that chyme is completely digested. Once it's digested, it's moved along into the other parts of the small intestine for absorption, and now the duodenum is empty, and everything that was inhibiting the gastric phase is now stopped, so that the gastric phase can start over again. And the gastric cell phase will begin over again, and another small portion of this will be sent to the duodenum, and that will begin the intestinal phase. So we go back and forth between gastric and intestinal phase for hours until the contents of the stomach are completely digested. Okay, so have an idea what's in each one of these phases. What are the secretions? This is going over the parts of the small intestine. This is the duodenum right here. And notice the relationship of the duodenum with other organs. Obviously, this part right here will be in contact with the stomach, but notice how close the duodenum is to the pancreas, which is the main organ secreting digestive enzymes, to the liver, with the gallbladder, which is secreting bile. So there's definitely a close connection between bile secretion, digestive enzyme secretion, and the duodenum. And we'll talk more about this in here in just a second. The small intestine has epithelium is folded, and these are permanent folds called plica. The folds are made of further folds called villi. So these are the little folds here are the villi, and these are all permanent folds. So this is one villi right here. So it will be one of these little finger-like projections right there is one villi. And then if you look at the cells, each individual cell has in turn folds. So this is fold, unfold, unfold, unfold. And what this is doing is increasing the surface area for absorption and digestion. So this is the trick that nature uses to increase surface area, is to fold surfaces. Okay, so the small intestine also has glands and crypts, absorptive cells that are going to absorb nutrients, goblet cells that will secrete mucus, enteroendocrine cells that are going to secrete enzymes like cholecystokinin and secretin. The cells, the simple columnar cells of the duodenum also secrete enzymes called brush border enzymes. These are digestive enzymes. So digestive enzymes are going to come from these epithelial cells lining the intestines, and they're also going to come from the pancreas, pancreatic digestive enzymes. Duodenal glands are going to secrete buffers to try to make the contents of the stomach, which are so acidic, to buffer them. The duodenum is not made to withstand acidity, so the minute it arrives has to be buffered. And then the contents, the chyme is moved, once it's been digested, is moved to the ilium and jejunum, where most of the absorption is going to take place, especially in the jejunum and the duodenum. Peyer's patches are found all throughout, but especially in the ilium, and Peyer's patches are loaded with immune system cells. The ilium joins with the cecum of the large intestine and the ileocecal sphincter and valve that move the contents along into the large intestine. Okay, so secretions in the small intestine, you have the mucus. We have secretin and cholecystokinin, which are hormones, brush border enzymes, pancreatic enzymes, and bile. So all these things are going to be secreted into the duodenum. Movements, peristalsis, and segmentation. Okay, so what we're going to do is we're going to stop here, and we have about 20 PowerPoints left. We will wrap it up next time, okay? And that way it doesn't make it into such a heavy lecture here. So next time we will wrap up digestion and we'll start with urinary system, okay? So I'm going to go ahead and sign off.